I hope you're doing well. For this next lecture, we're going to focus on a couple of cell structures, primarily one, bacterial cell wall, but we will jump into another one that is somewhat related as well. So this lecture will be a bit shorter, but it'll be an extension of what we were talking about before, which is a focus on some specific cell structures and the molecules that make them up, and how they can sometimes influence bacteria causing disease, in the case of being a virulence factor, but also just the roles that they play for the cells that we're talking about. So in this lecture, we're going to focus on bacteria. So we'll be looking at bacterial cell walls and specifically two types of bacterial cell walls. But I do want to highlight that cell walls are found on the exterior of multiple different types of cells. So plants have cell walls, fungi have cell walls, uh, bacteria have cell walls, and all of these structures are made of different molecules. So in plants, the external structure, their cell wall, is made of cellulose that you may have learned about before. Cellulose is actually the reason why we can't digest grass as humans and why it can make you sick. Fungi have chitin as the molecule that builds their cell wall. And in bacteria, we have a unique molecule known as peptidoglycan that is present in most bacterial cell walls and the two types that we will focus on. If we break down the word peptidoglycan, you probably are thinking about two different halves of the word, peptido and glycan. And if you think about that peptido, what is a word that you may have heard previously that's similar to that? And same thing for glycan. What's a word that's similar to that? If you know, jot it down. If not, no worries. But peptido is very similar to peptide, and glycan is very similar to glyco, which means sugar or glucose. And so peptidoglycan, when taken together, is like a peptide combined with sugar. Peptides are like small proteins, and sugars are macromolecules that are important for a variety of things, oftentimes structure or energy. And in the case of peptidoglycan, we have a combination of molecules that make both of them to make this larger overall molecule. There are stretches or chains, sorry, of alternating sugars called NAG and NAM that run, essentially you can think of it as in parallel to each other. And those chains of sugars, or those linked sugars, are then connected by amino acids, which form these peptides or these small protein linkages. And so what you end up with is sugars running in one direction, and those chains of sugars being linked by short proteins or combinations of amino acids to form a mesh-like structure. So some going this way, connected going this way, you end up with this overall mesh and this overall rigid structure. And so the way that I think about peptidoglycan and how it goes into forming the cell wall is you have this layer of a molecule surrounding the cell that functions as sort of a cage. So you end up with a cage-like structure surrounding the cell that provides it with some strength, some support, and some structure. We'll talk about why that's important here in a second. If we zoom in, you can take a look at the structure that I was describing even more closely here. So you see the NAG and NAM carbohydrates alternating and then linked by those peptides or amino acids that run in the perpendicular direction. So we get this overall mesh-like structure, this rigid-like strong structure that goes into forming the cell wall. And if we take another look at it, this isn't an organic chemistry class, and I don't expect you to know all of these individual structures. But if you take a look at what the molecules themselves actually look like at the atomic level, when we draw out the structures, you can see that we have sugars that are connected to each other. These would continue to be connected in that carbohydrate chain. And then you can see the amino acids that branch out in order to form that peptide cross bridge or connecting to another chain elsewhere. Okay, so think that's the molecule, peptidoglycan, that goes into making the cell wall. Let's take a step back and think about, why does it make sense for a bacterial cell to have a cage-like structure surrounding it, to have that physical support, that rigid structure surrounding the entirety of the cell? And I would guess that if you come up with an answer in thinking about this, you probably are going to say some version of protection. Maybe not, but most people, when I ask this question, say, oh, well, it gives you protection. You're surrounding the cell with a strong structure. That's going to protect the cell. So I want to actually ask a follow-up instead of spending time on you thinking about that. What does the cell wall protect the cell from? And there are technically a couple of answers, but I want you to think of mostly, or primarily, what does the cell wall protect the cell from? Okay, now that you've thought about it, most people tend to say probably protection from harm on the outside of the cell. And that is technically true, but that's not really the main thing that the cell is protected from. So I want you to keep that in mind, because we're going to take a look at a concept here. And it's important to note that it's not actually protecting the cell from what's going on the outside. Okay, so if it's not protecting the cell from what's occurring on the outside of the cell, then what must it be protecting the cell from? What's going on the inside? Okay, what is going on the inside of the cell? Well, a lot of things are going on the inside of the cell, but they're all things that the cell wants to carry out, all jobs that the cell wants to accomplish. So why would it need protection from any of those things? And that brings us to a concept that we need to make sure we're familiar with, that's known as osmotic pressure, and a related concept known as Turgor pressure. Okay, I want you for a second to just picture a water balloon. Imagine you have a water balloon and you are getting ready to go into a water balloon fight. And so you hook the water balloon onto a hose and you turn it on and you let that water balloon fill with water that's entering into the balloon. Okay, what's going to actually happen to the balloon itself physically? As you know, it's going to expand, right? And let's say we keep it on the hose. What's going to continue to happen? It's going to expand even more. Then we leave it on. It's going to expand even more. Then we leave it on. It's going to expand even more. And eventually, what is going to happen if we never stop the flow of water into the cell? Or sorry, into the balloon? It's going to burst, right? The water within that balloon is going to apply enough pressure on the walls of the balloon that eventually it'll be too strong of a force. The balloon won't be able to withstand it, and it'll burst open. And the contents of that balloon, the water, will spill out and go everywhere. Now we have a non-functional balloon, right? It doesn't serve its purpose anymore. That's because of osmotic pressure that's occurring. And technically, the bursting is because of Turgor pressure. Turgor pressure refers to pressure of water molecules hitting the side of a boundary. In the case of a biological membrane, hitting the cell membrane because there are so many water molecules moving around, right? You can imagine if a cell were to fill up more and more with water, there are going to be more and more water molecules bumping into each other and able to bump into that cell membrane and pushing out on it, causing stress. Osmotic pressure is the idea that there are forces that will drive water in a specific direction, either into the cell or out of the cell. And this has to do with the idea that the cell membrane is a semi-permeable membrane, meaning some things can pass through and other things cannot. In our case, what we're focused on is the fact that water can pass through, but solutes, so things like salt or sugar or other stuff, can't just readily pass back and forth. it's stuck on one side of the membrane or the other. That makes sense. You don't want things to be able to just mix with the outside world. That defeats the purpose of having a membrane. The membrane's there to separate the cell from the environment. But water can generally flow freely in between because of proteins that allow it to within the membrane, and salt largely is stuck on one side or the other. Okay, so let's imagine, first of all, that we have a cell that's just in standard state. So the internal concentration of stuff, of solutes, salt, proteins, sugars, everything, is 0.75%. If the concentration of solutes, or of stuff that's not water, is the same on the outside of the cell, so also 0.75%, then water will pass back and forth across the membrane, but it'll do so equally. And as a result, the cell's not going to change its size at all. There's going to be an equal rate of water coming into the cell as what is leaving the cell. And so if we're just viewing it, if we were able to take a video of it and just watch it, it would look essentially like nothing was happening. Technically, water is going back and forth, but really the cell is just maintaining its state. Okay, but now let's say that we take a cell and we put it into a container that has pure water. So there is 0% solutes in there. Okay, now what's going to happen? Now, you probably learned this in another course, but we want to think about where water is going to flow as a result of osmotic pressure. If there is no salt outside of the cell and some salt inside of the cell, the water is going to try to balance out the concentration of salt on both sides of that membrane. And so water overall will flow toward a higher solute concentration, or toward the salt. So in this case, if you put a cell that has stuff within it, has solutes within it, in pure water that doesn't have stuff within it, the water is going to flow into the cell, making an attempt to balance the concentrations out. And in doing so, what is going to happen to the cell itself? Well, if water's flowing in, it's going to be like that balloon that we saw. It's going to keep expanding in size. It's going to get bigger and bigger and bigger. Okay, now let's look at the opposite situation. Let's say you have a cell that has 0.75% solutes on the inside, and you put it into a solution of salt water that now has 1.5% solutes on the outside. So double. There's twice as much stuff outside the cell as there is inside the cell. Which direction is water going to flow? Water is going to flow toward the higher stuff concentration, toward the higher solute concentration. So now water is going to flow out of the cell to try to balance those concentrations. As a result, you're going to end up with the cell losing size. Right? If it's losing water, you can imagine some of the stuff that's filling up the cell is leaving, going outside, it's going to shrivel. You're going to see it shrink. Okay, this presents a unique obstacle or challenge for a cell living in environmental situations, because a cell can't guarantee that it's always going to be in a solution that has an equal amount of solutes outside of the cell compared to what it currently has going on in the cell. And so as a result, it needs to be able to protect against major changes in osmotic pressure, where you would see water really flow into the cell, causing it to swell up and grow in size and ultimately potentially burst, or flow out of the cell and shrivel up, where the cell could become dehydrated and may run into problems for the opposite reason. The cell wall offers protection from what's known as osmotic lysis, which is the situation where water's flowing into the cell and you are threatening to have it burst. You're getting an increase in turgor pressure. The membrane's getting a lot of stress on it because the cell keeps expanding and expanding and expanding, and eventually it could burst. Okay, think about what we said the cell wall was. It was strong, it was rigid, it was a structure that was actually surrounding the cell, acting sort of like a cage. If you were to put a cage around a water balloon and fill it up with water, eventually when it gets to the size of that cage, the water's not going to enter into the balloon nearly as easily, right? Because now the cage pushes back. That's essentially what the cell wall does. It provides structure, it provides strength, so that in the case of changing osmotic pressure, the cell can avoid osmotic lysis. Even if it swells, the membrane will swell up to the cell wall, but then that cell wall will provide some strength and structure to prevent it from continuing and ultimately bursting. When a cell bursts, it's a process known as lysis. And when a cell lyses, it's significant to the cell because that cell is now dead. If you think about it, we have these highly organized cells that have a bunch of stuff within them doing very specific roles for life. If a membrane breaks open and now everything just pours out, that cell no longer exists. It's now just mixing with its environment. It's a dead cell. So the primary purpose of a cell wall for bacteria is to avoid osmotic lysis by being able to oppose increasing turgor pressure in the case of a cell swelling when it's put into a solution of a lower solute concentration. We can actually take advantage of the fact that this can happen to cells in treating certain bacteria in ways to kill them to avoid infection. You have likely heard of penicillin. We will talk about penicillin specifically in the future. But for those of you that aren't familiar with it, penicillin is an antibiotic, so it kills bacterial cells. And it does so by preventing cross-link formation in peptidoglycan. So if you think back to that structure that we talked about, we were creating a cage by having carbohydrates run in one direction and peptides linking those together running in the other direction. Penicillin prevents key steps in the synthesis of peptidoglycan that results in the cage essentially not being able to be finished. So when you take penicillin, you are taking a molecule that will interact with bacteria that could be present in your body, and they will not be able to effectively create or synthesize peptidoglycan. As a result, they will make a very weak cell wall or a very incomplete cell wall. And now all of the issues we just talked about regarding osmotic lysis come into play. The cell now becomes much more likely to burst or undergo osmotic lysis. Our bodies also make a protein known as lysozyme, which is an enzyme that digests intact peptidoglycan. So now this is a little bit different from penicillin. Keep in mind, penicillin was a molecule that disrupted making peptidoglycan. Lysozyme is a protein that we make as a part of our immune system that will act as scissors to cut peptidoglycan that has already been made. So it will find the peptidoglycan and chop it up into smaller pieces. The result is, again, a weakened cell wall, because now that rigid, strong structure that's helping to prevent the cell from undergoing osmotic lysis is weakened, is broken. And again, if the cell encounters a situation where it swells at all, which it's probably going to in its lifetime, it now risks this osmotic lysis. It'll burst open and we will see it die. And in fact, if you take a look at the picture here on the right, you can see here is a cell that has an ineffective cell wall. And so what is happening to the cell? Well, if you take a look at it, the contents are spilling out, right? It's erupted up at the top. It's burst up at the top. The internal contents are spilling out. This is a lysed cell. The cell membrane has burst open. Since it's a lysed cell, how else can we describe it? We can describe it as a dead cell. And so this is why lysozyme is something really important in our immune system and why penicillin was such a big discovery in the world of antibiotics. Okay, so now we're going to talk about a couple of types of cell walls, the most common types that we will encounter in our course. So we'll focus on two. I want to make note these aren't the only two types of bacterial cell walls or outer boundaries that you see or outer coverings that you see, but they're the most common types and they're what we'll focus on in our course. And they're termed gram-positive and gram-negative cell walls. You will encounter the terms gram-positive and gram-negative quite frequently in this course because it's a really common way that we distinguish between different bacteria, different species. Some species are gram-positive, some are gram-negative. They show up differently when you carry out what's known as a gram stain, and which you will learn how to do in lab. And that is a key way that we can indicate what type of bacteria we're taking a look at. We're going to start by focusing on a gram-positive cell wall, which overall I would describe as a simpler version of a cell wall, but still really effective at preventing osmotic lysis. In fact, the most effective type of cell wall when it comes to preventing osmotic lysis in bacteria. If we take a look at this cartoon over on the left, we can see the parts that go into making the cell wall. So we'll start down at the bottom just so we can orient ourselves in terms of what we're looking at. We have the cytoplasm, which is the interior of the bacterial cell. That's going to be surrounded by the plasma membrane or the cell membrane that we see here. So we have the lipid bilayer forming that outer boundary of the cell. On top of that, so outside of that, is where we actually see the gram-positive cell wall. And you can see here that there's just a few distinct parts that make up that gram-positive cell wall. First, in blue, you'll see multiple layers or a very thick layer of the molecule peptidoglycan. So we talked about peptidoglycan and looked at those structures earlier. This is going to be that cage-like structure, that overall mesh-like structure that provides strength. That's going to be sitting right on the outside of the plasma membrane, but it does need to be anchored to the membrane in some way so that it stays connected. And that is done through molecules that extend from the plasma membrane that are known as teichoic acids, which we see labeled here. Teichoic acids have a minor negative charge, so overall their primary role is to connect the thick peptidoglycan to the plasma membrane and give a little bit of negative charge to the very outside of the cell. This negative charge can be helpful because it will attract positive charges that the cell needs to come closer. It will repel negative charges that will push things away that the cell doesn't want to be close to. But overall, it's mostly just to help anchor the cell wall structure to the gram-positive cell. And so you notice that this is quite simple. But if you think about it, a very thick, strong cage-like structure is something that's going to provide a lot of resistance to oppose osmotic lysis. If that cell is swelling, the plasma membrane keeps expanding out and out and out, a thick layer of peptidoglycan is going to push back to prevent the cell from ultimately lysing. Okay. Next, we want to take a look at a gram-negative cell wall. And if you take a look at the pictures here, just glancing at them, you will see that a gram-negative cell wall has a bit more going on. There's more to it. There's more labels just in general. And the primary reason for that is because a gram-negative cell wall has a separate outer membrane. So now, rather than just a thick layer of peptidoglycan and just a simple cage, a gram-negative cell wall has a thin layer of peptidoglycan that is anchored to an outer membrane, even outside of that peptidoglycan. And so what you have now is an added layer of protection from things on the outside with a smaller layer of protection from peptidoglycan to resist osmotic lysis. It still helps with that, but not quite as effectively as that thick layer we saw in the gram-positive cell wall. Something unique to a gram-negative cell wall that we will talk more about and that you'll come across quite a bit is a molecule known as lipopolysaccharide, or LPS. And LPS is the lipid, the sort of sugar lipid, that extends out from the outer membrane. So we have the very inside of the cell, we have the plasma membrane, so similar to what we saw before. We then have a thin layer of peptidoglycan attached to a second outer membrane, and the very outer layer of that outer membrane is made of LPS. So LPS is something that is specific to gram-negative bacteria, and it's something that plays a crucial role overall in helping the gram-negative cell wall to achieve some added protection from the outside world. If you take a look at this second cartoon here, you can see that the LPS here on the very outside of the membrane has long stretches of carbohydrates, long stretches of sugars that extend out into the surrounding environment. And those sugars overall have a net negative charge. Now you may recall on the gram-positive cell wall that teichoic acids had a slight negative charge. These sugars carry a more significant negative charge, meaning that you get more repulsion of negative charges, more attraction of positive charges for things going by the cell, and also the fact that we have this construction of a second outer membrane, a whole second barrier. More things passing by will have a harder time actually getting into the cell and interacting with it. So gram-negative cells overall, because of their cell wall, have added protection from the outside world. Still have some protection from osmotic lysis, but not to the same extent as the gram-positive cells, but then more protection from things on the outside world. Here's just a couple more pictures. I always like to include as many pictures as possible so that if you prefer to take a look at these when you go back and are studying or keeping track of what makes up the cell wall, you can see the different layers, you can see the multiple membranes. All of these are giving the same overall idea of what makes up a gram-negative cell wall. So whichever one works best for you is what I recommend referring to. I want to zoom in a little bit more on that LPS molecule that makes up the very outer leaflet or the very outer part of the outer membrane of the gram-negative cell wall. LPS is a lipid that has a bunch of sugars attached to it. So lipopolysaccharide, polysaccharide meaning many sugars, lipo meaning lipid. What we see is that LPS as a molecule can be divided into three separate regions. We have the part that actually sticks into the membrane, which is known as lipid A. Makes sense. It's very similar to lipids that we traditionally see in membranes. That's what's going to anchor the LPS in the outer membrane. Attached to that directly is what's known as the core polysaccharide, and the core polysaccharide is always the region that has sugars that are always linking the very outside of LPS to lipid A sticking down in the membrane. And then the outermost portion of LPS is what's known as the O-specific polysaccharide or the O antigen. And the O region of LPS is unique to different species of bacteria. So you'll have different lengths, different combinations of sugar that make this up, giving different species slightly different characteristics, different strains of bacteria, even slightly different characteristics based on the composition of that outer part of their cell wall. And it turns out that this is really important when we're talking about human health, because overall LPS can be recognized by our immune system. So we can detect the presence of gram-negative bacteria if they happen to be present, but also it can significantly activate the immune system. Now, what's interesting about this is the part of LPS that stimulates our immune system. It may not be what you think. It's not the outermost part. It's not the O antigen. It's actually lipid A, the part of LPS that sticks down into the outer membrane. So take a second and think about it. Why would lipid A, a part of LPS that normally isn't even exposed to the outside world, be something that our body can even detect? How would we sense that? Okay, you may have come up with it, you may not have, but if a cell lyses, so if a cell dies and the outer membrane and the cell wall of that gram-negative bacteria falls apart, now you have lipid A free floating in the environment, which means that our body can detect it, and it stimulates a really, really significant immune response, one that is good, but if it's out of control can cause some major problems in terms of disease. There is a specific strain of E. coli that has a type of LPS that is associated with disease. It's not actually the LPS itself that's causing this disease, but you can detect that you have that strain of pathogenic E. coli. I believe I said in an earlier lecture, you have more E. coli cells in you than human cells. It's very commonly a part of you. It's not something that normally causes disease. However, there are some strains of E. coli that can cause issues. One of them is E. coli 0157H7. A specific marker that identifies that E. coli is the unique O antigen on its LPS. So not something that you need to memorize necessarily, but just to be aware of in the unique nature of LPS and the effects that it can have in terms of detection. Okay, just quick review again. If you take a look, we have the O antigen in red over here. We have the core polysaccharide, which is that linking region in the middle of the LPS molecule, and then we have lipid A down at the bottom, which is what actually sticks into the outer membrane. And just as a reminder, it is that lipid A that actually is something that our immune system can react to, and sometimes that will cause a problem for us. Okay, so how can it cause a problem for us? The way that it can cause a problem is when a cell breaks down. So over here in this animation, you can see the death of a gram-negative bacterial cell. You see that it lyses, it falls apart, and some of that lipid A, known as endotoxin, is being released from the cell. And now our body can interact with it. When our body detects the presence of high levels of lipid A or endotoxin, we get a really exaggerated inflammation response. And we'll talk more about this in Unit 4, but it's good for us to have that inflammation response. But if it goes overboard, then we start to damage our own tissues. And as a result, we can experience a type of shock syndrome where our body goes into shock, destroying itself in an immune response to a molecule that it's not expecting to be there. Okay, so I want you to think about, and we're going to tie this back to something we talked about previously. When we're talking about lipid A or LPS, let's talk about LPS as a whole, even though it is lipid A here specifically that's endotoxin. If we're talking about LPS as a whole, would you describe that as a cell structure, or as a molecule? We know it's a physical thing. How would you characterize it in terms of organization, the levels that we've talked about? Take a second and think about it. Okay, we know it's not a cell because it's not the entire thing. It's not the entire bacterial cell. It's not a cell structure because it's a building block of a cell structure. In this case, the cell wall. It makes up a part of the outer membrane of a gram-negative cell wall. LPS is a molecule. Okay, now in the past, when we talked about virulence factors, we said if we have a cell structure or a molecule that ends up helping to cause a disease state, we have a virulence factor. So would you describe LPS as a virulence factor? And yes, we would, right? It's a molecule, so it fits under that category, cell structure or molecule. It's a molecule. And does it lead to a disease state in us? Yeah, absolutely, right? We can have that exaggerated inflammation response that leads to tissue damage, which is a negative state of health. LPS is a really clean example of a virulence factor that you find in gram-negative bacteria. Okay, so just quickly, I want to point out here on the left, we have a gram-positive cell wall, and on the right, we have a gram-negative cell wall. It's important to understand the differences between the two. Just as a quick reminder, in the gram-positive cell wall, we have a thick layer of peptidoglycan. In the gram-negative cell wall, we have a very thin layer of peptidoglycan. On the gram-positive cell wall, we see teichoic acid present that serves as an anchor of the peptidoglycan to the plasma membrane. The gram-negative cell wall does not use teichoic acid. The gram-negative cell wall has an outer membrane, and the gram-positive cell wall does not. And within that gram-negative outer membrane, the molecule LPS forms the very outer leaflet and sticks out with that negative charge to the surrounding environment. And gram-positive bacteria do not have LPS, don't have an outer membrane, don't have LPS, don't have that higher negative charge sticking out into the environment. So I just compared and contrasted physically gram-positive and gram-negative cell walls. Why is this something that is important medically or environmentally or in terms of us diagnosing a potential pathogen? Well, in the case of medical significance, this becomes really, really important because if we are going to target the cell wall as a way that we're going to try to weaken a pathogen, similar to like we saw in that image earlier, the lysing bacterial cell, we need to make sure we're using a drug that specifically targets the type of cell wall that we want to target. If you currently have an infection with a gram-positive bacteria and you target a gram-negative cell wall, so maybe something to do with LPS, is it going to be effective? No, because your target doesn't have what you can actually target. So gram-positive and gram-negative bacteria do respond differently to different antibiotics because of the nature of their cell wall, especially if we're targeting that cell wall. So the medical significance is you really want to know what you are targeting before you take steps to specifically try to get rid of it. In terms of environmental significance, I want to actually pose the question to you. If we were to put each of these types of cells, so a gram-positive cell and a gram-negative cell, into pure water, allowing the net flow of water to go into the cell, causing swelling, which one do you expect would be better at resisting osmotic lysis? Take a second and think about it. Remember, we're talking about water pushing on the plasma membrane and ultimately having a chance of lysing the cell, which would kill it. Which of these would survive that better? And I hinted at this earlier, but the answer here is the gram-positive cell wall. Right? That thick, really strong layer of peptidoglycan is going to provide more support and more strength to resist osmotic lysis. Gram-negative still has some peptidoglycan to assist with that, but not nearly to the same extent. We can just see it here in the drawing on the right. Layer A, the peptidoglycan, much, much thicker in a gram-positive cell wall. And then finally, what's the diagnostic significance? This is really similar to the medical significance. You want to be able to determine what it is that you actually are trying to get rid of if you have a patient with an infection or if you have an infection. And so making sure that you use an appropriate technique to determine what type of cell wall the bacteria has can help you narrow in on the appropriate treatment to use. Okay, some things to keep in mind when you're looking back at the cell wall. Can you explain which one survives better in the environment? We just talked about that. Can you explain the medical significance? We just talked about that. Just as a hint overall, or a word of encouragement in terms of your studying, I would try to make your explanations relatively short, but make sure they're very clear and that they do cover everything. You want to be specific, you want to be clear. That way you're really identifying the key differences between the two and what that means for the cell overall. All right, the last type of cell structure that we're going to talk about in this lecture that is peripherally related to the cell wall, but isn't the same as the cell wall, is what's known as an endospore. And an endospore is a really unique structure that is made only by certain bacteria. And in fact, the bacteria that can make these endospores are always gram-positive. So that's where you have a relation to the cell wall. An endospore is a structure that allows for a bacterial cell to package away some of its DNA and other materials into a structure that's really, really strong, really, really resilient, and able to survive exceptionally harsh conditions. That way, when the cell itself dies because the conditions aren't very favorable, it's not actually being lost forever, because the endospore will stick around and continue to store the DNA, the RNA, and proteins that the cell set aside for better conditions. The way this works is these species of bacteria will make a copy of their DNA, just like anything would when it's reproducing. But instead of forming a whole new cell, they package that DNA by wrapping it in a part of their plasma membrane. Ends up being wrapped in 2 layers of membrane and then further packaged with a surrounding coat of peptidoglycan, so related to that cell wall that we saw before. What's really unique about the endospore is it has a very, very low water content. So it has DNA, RNA, protein, a lot of things that are necessary within it that are similar to the contents of a cell, but not very much water. And what this allows the cell to do is, let's say that conditions get really bad, everything dries out and the cell dehydrates. So ultimately it dies, and as a result it lyses, loses its structure and bursts open. That endospore that's been packaged with all of those key pieces of the cell gets released to the environment, but now it's resistant to those really, really bad conditions. It's not actively a cell. It's not technically fully alive. Endospores are what are known as pseudo-life or pseudo-living because it's sort of a halfway point. And it'll just continue to exist as a package for later. When conditions improve, in this case, water comes back, the endospore will rehydrate and it can convert into a fully viable cell. What that allows for is a species of bacteria to survive a really dramatic shift in harsh conditions and ultimately continue to live even years past when those harsh conditions have occurred. In fact, research has found endospores that are hundreds of thousands to millions of years old that are still able to turn into fully functional viable cells. So this is a real long-term strategy for certain species of bacteria to be able to get through really, really tough conditions. Those conditions are usually heat and dryness, but sometimes certain toxic materials, acids or bases, certain disinfectants, and then also notably radiation, including sun exposure. This is not binary fission. This isn't a cell fully reproducing. It's not making an exact copy of itself, but it's setting aside key parts that are needed for a new cell to emerge when times get better. When we previously talked about Robert Koch and anthrax, the part of anthrax that actually serves as a virulence factor that ultimately causes disease is an endospore. So it's the endospores that actually make up anthrax and cause the really detrimental effects on humans or other mammals. And here's a picture of some endospores. This is the same figure as before, just walking you through the whole process, just zoomed in a little bit more. And then I did want to highlight for you here that there's an electron micrograph, so you can see the dense nature of all of those things packaged within the endospore within the larger cell. And here is what's known as an endospore stain. We will not do it in lab because this contains some chemicals that are pretty toxic that you have to use. But if you look, all of the pink stained cells here are living cells, and those green structures are stained endospores. So you can see that some of them have been released from cells. Everything in this picture is from the same species. It's just whether it's a current viable cell or an endospore that you are looking at. In some of the cells, you can even see the white dot that's present. That is the endospore that is still existing within the living cell. It hasn't yet been released because that cell is still alive and thriving. All right, and we will call it there for the cell wall and endospores and wrapping up overall what we've talked about with cell structures and molecules. So I hope you have a great rest of your day, and I will see you in the next lecture.